And those are genetically distinct breeding populations. So one of them has an anadromous life history, the other has a resident life history. The difference between them is this one spawns in the lake, this one spawns in the river. This one reproduces generally once, this can reproduce up to 7 times, just like a brown trout can. So we've had a reversion back to the brown trout life history. Here's another one that's even more interesting. You go to a little stream here on the end of Newfoundland. It's called Cape Brace in this area here. This is a fully mature Atlantic salmon female, 12 centimeters long. You can see the vent there. These are the eggs it's produced. It produced 60 eggs. That's as old as it gets. Average lifespan, 2 years old. Males, females, exactly the same. It looks just like a salmon parr, but that's as far as it gets. I caught this fish about 50, 60 meters from the sea. It could have gone to sea. It doesn't go to sea. Why doesn't it go to sea? Well, its ancestors did, but it's evolved to become a resident Atlantic salmon population. There are no brown trout in Newfoundland. Native brown trout. They have been introduced, but here we get variation in adult run timing in the River Tay in Scotland, and what you have is a lot of variation in the altitude of streams, the River Tummel in the Tay and the River Almond. And Davy Stewart at our laboratory and others, they did a comparison to look at the heritability of run timing, and they used the kind of experimental approach that Andy mentioned, basically a common garden experiment. They looked at the run timing of the adult native fish, the 2C winter fish versus a 1C winter fish. And basically from the Tummel, the 2C winter fish come in early, and the 1C winter fish come in later, but when you compare them to the River Almond, you see that the 2C winter fish in the Tummel are earlier than the 2C winter fish in the Almond, and the 1C winter fish in the Tummel are earlier than the 1C winter fish in the Almond. Now, is this an environmental effect, or is that a heritable effect? Well, what they did was they transferred the fish from these two systems in this transplant experiment into the River Bran there, which doesn't have a native population at all. There's a waterfall there. And what they found when they transferred them was there was a retention of this timing difference in the two stocks. But as you'll notice, it's not as marked. So we have again a genotype-environment interaction here between the run timing. Some of it's heritable, some of it's environmentally determined. Oops. Sorry, just go to that. They also did a study on smolt run timing with the same populations. They find timing differences, and again, you have this genotype-environment interaction. There are heritable differences between the salmon in these two parts of the system. So homing, as Andy mentioned, really is the key to what's going on here. And this makes the fact that the salmon, like the trout, are actually segregated or restricted into these components we call breeding populations, within which you get the animals breed at more or less at random, but among which you have very highly constrained gene flow going on. So the thing that we're very interested in is defining how a river stock of Atlantic salmon is actually divided into breeding populations. Is it a single breeding population in every river system? Is it 2 populations? Is it 100 populations? This is actually terribly important from a management perspective. Do you have 1 population to manage, or do you have 100 that you've got to manage? So, do you have 2? Is it really highly subdivided? So the research we've been doing is very much focusing on trying to understand that structuring that's going on. However, if you think about it, it's incredibly difficult to define spatial boundaries. If you tried to define the spatial boundaries of human breeding populations way back before people like me traveled all over the world, and my ancestors did as well, it would have been very difficult. I remember, I studied at Nottingham University not far from here, and I remember there was 3 changes in accents between Nottingham and Derby. And I swear there couldn't have been an awful lot of reproductive mixing as well, because there were very marked differences culturally in that very short distance. But if you had to define the breeding population of Nottingham as opposed to the one in Derby, and where the boundaries of these are actually defined, it would be incredibly difficult, even if you went and looked at church records and you talked to people. But you try and do it with fish. That's an incredible challenge. So what do we use? As Andy mentioned, we go into these molecular genetics and we try and understand the genome, and we all have these heritable differences among individuals, among populations, etc. If we track these, just like you use in human DNA forensics to do paternity analyses or to find criminals in rape cases or burglary or whatever, you can do exactly the same thing in fish. It's the same basic principle. The only difference in salmonids is here, these are the chromosomes of an Atlantic salmon, and there's 58 of them if you happen to be a European salmon, and there's 56 of them if you happen to be a North American salmon. And in trout, they've got 84 of these in some populations. I think there's lots of variation going on there as well. But the important point is that this genome is twice as big as the human genome. So we think we're quite sophisticated. We've got about 4 billion base pairs. There's upwards of about 8 billion base pairs in this genome. So that's a huge amount of information when you think about it. And this unravels until you get all these base pairs here, and then we read those base pairs just like you do a book. But Andy mentioned we can look at several thousand genes, but that's still a fraction of a fraction of a fraction of 1% of the genome of an Atlantic salmon that we're looking at. And all the rest of it we don't know anything about yet. But we can do it, and I won't go into the methods here. There's various ways of reading little parts of this. We use various tricks that have been developed as part of the Human Genome Program, and then we read those little parts, and we get all sorts of fancy things out of it like this. It's like reading tea leaves a bit. However, the important point is how we interpret it. And it's a little bit like computers. You know, computers are made just of gates. They're either on or they're off. But when you think about a computer, what you can do with a computer, I mean, just the mere fact that you can produce a graphic like this with just little switches that go on and off is quite impressive. You wouldn't have imagined that just from the concept of on and off. Well, the genome is similar. You've got 4 bases that just vary in their presence or absence and in their order, and we've got a genome that produces animals and life that goes on. It's the same complexity. So we're reading that basically. This is a study that was done on a very small set of rivers near Oban on the west coast of Scotland called the Fiachan Lochs. And you see the scale here is, this is about 15 kilometers here, so not very far, but a little bit further than between Nottingham and Derby. And this is the Euchre here. We took samples of salmon from this system and from the Nell Lonan system here in this small lower tributary of that system, the Fiachan Beak. And what we wanted to ask here was whether the salmon in this system were genetically different, and were they composed of different breeding populations in these different rivers, and if so, could we say where the fish that were being taken in this small fishery here, which rivers they came from? Because the owners here were in a bit of a tiff, because they were saying they were taking each other's fish, and they were blaming it. And they really wanted to know what the answer, whose fish were being taken. So we said, well, maybe we'll be able to help you out here. So we looked at that, and amazingly, what we found was that in every location that we looked here, there was a different breeding population of Atlantic salmon. So in 15 kilometers of river here, there's 3 distinct breeding populations of Atlantic salmon. Now, I wouldn't have imagined that the scale of it would have been that good. What we have here is a semi-passable falls. What we have here is a loch. So we got physical structuring and breaking up of the salmon stock in this river into these breeding populations, which is correlated with landscape features. And here it's a branching of the system, here's another loch dividing these two, etc. So we've got 6 breeding populations in an incredibly small area, and each one of those is contributing to the stock production in that area. So the genetic differences here were sufficient to allow us to actually tell where the origin of most of these fish that were caught there were due. But the thing that this particularly told us was that most river systems of Atlantic salmon are structured into multiple distinct breeding populations.